So, yeah, 20 years ago, my PhD supervisor, which was Jeff Hinton, said to me, Brendan, you're making things too complicated. Keep it simple. It was very good advice. It comes back to Occam's razor and sort of what we believe in terms of science, right? We're hoping to find something simple. So that's what I try to do. I try to approach scientific questions in the simplest fashion that I can. Of course, my perspective might be different from other people's perspective. Some people would say, well, the simplest thing to do is conduct an experiment. And I very much respect that. I really like Jay Shinjuri's talk and the experimental data that he's generating. I think it's just fabulous. And I'll be using that for sure. But of course, for me, I got to stick true to what where I was born and what I grew up, what I was taught, and that's computational simplicity and aspects of biology that for me resonate in terms of computational structure. So concepts like combinatorics, concepts like statistics, things like that, right? So that's been my perspective, and that's what I try to get my group members to think about all the time. I was talking to Ron yesterday. We were discussing about how people do research in biology and computational biology, and one of the issues is that new data sets are always being generated. So as computational researchers, one thing we're worried about is we solve a problem computationally, but maybe a few years down the road, someone's just going to make our result irrelevant because there will be a new experimental technology. So what I try to think about is, if I, as a computational researcher, have something valuable to add, it should not be a 3-year timeline, right? It should be something that I think might last 20 years or 50 years. So I keep that in mind. I believe in the things I know about as a computational researcher, that they are valuable in biology. I could be wrong, right? Could be completely wrong. Maybe in the end, computation will be irrelevant. In a decade, or in 100 years, people look back on this time and say, wow, that computational stuff was kind of a distraction. But I don't believe that, and so I'm going to stick to my guns. All right, that's enough of I think that's valuable, philosophy, but enough of the philosophy. Of course, none of the work that I'm going to talk about today could have been done without my group members, and I'll talk about them later on. I should also add that this is my first time here, and I very much thank the organizers for inviting me, because I feel like I found my home. I haven't gone to many conferences. As a lot of people know in this room, I travel to universities and give talks because I like to see people and talk to people one-on-one. I've avoided conferences because most conferences just haven't fit my interests, but actually I've really enjoyed the last few days. So I think this is just a terrific conference in terms of match to my interests. So I'd like to address all sorts of things that people have talked about, like the single cell analysis that I was talking about yesterday. Lots of other things, but unfortunately I won't be able to do that in my talk. So let me just go back to what Nancy was talking about. I just wanted to make a comment, and I discussed this with her yesterday before she left. She said, we're not finding large effects, right? And she showed this picture, which I thought was a great, a really nice analogy. We're not seeing these giant buildings, okay? And so this is addressing the question of large effects versus many small effects. Well, perhaps it's simply because we're looking at the picture this way, right? If you're looking at the picture this way, then yes, you don't see the big effects. Doesn't mean they don't exist, but they're there. You just don't see them. So maybe we just need to look at things differently. And I think really that's what this group is all about, right? Using regulatory models to look at things differently. So I like to give people an indication, maybe this is not so relevant to this group. I like to give people an indication of the importance of regulatory models. If we look at the landscape of damaging mutations, we of course know that a mutation that causes a missense or maybe a stop code on introduction or something like that is problematic, potentially. But there's another kind of mutation, which is a mutation that affects regulatory code. All right, sequences within DNA that turn on genes, turn them on and off, control splicing, control polyadenylation, things like that. This is a much larger space, and that's the focus of my group. Okay. So my group is developing techniques based on regulatory models to score the impact of all mutations that affect regulation. And I'll, of course, be talking about this for the rest of my presentation, but Just to give you an idea, we have a system that can score mutations based on regulatory models. If we look at the overlap here, so these are mutations within exons that have an effect on regulation. We can look at synonymous mutations. And if we look at synonymous mutations, we find that those are 10 times more likely to be implicated in disease than common SNPs when we use our system for scoring them. Okay, so we can score synonymous mutations. And when we do that, we actually find that they're very, very enriched for disease annotations. Now, even if you look at missense mutations, you might think, well, missense, of course, that's going to change the protein. If you look at missense mutations that are in unstructured protein domains, unstructured regions of proteins, so they likely don't have much of a functional effect on the protein, we find that those mutations are 5 times more likely to have a high regulatory score using our system compared to common mutations. Okay, so even for missense mutations, we're able to dig up some interesting results. Okay, so the specific aims are as follows. We want to introduce computational frameworks for inferring these regulatory models. We want to generate tools that can be used by the community, and we have online tools that you can use. They're getting better and better, predicting expression essentially, or mRNA levels from DNA sequence, and then of course identifying and scoring genetic variants, and then finally understanding and diagnosing genetic diseases. And this is something I wasn't doing 2 years ago, but now that we have the system up and running, we're collaborating with a variety of different hospitals to explore different diseases, pancreatic cancer with Lincoln Stein, and autism spectrum disorder with Steve Scher, and several other examples. All right, I wanted to dig into one example before I start, because as computational people, we often talk in generalities. So let's talk about the genetics of spinal muscular atrophy. How many people are familiar with the genetics of spinal muscular atrophy? Raise your hands. Okay. Great. So after today, you will be. At least, well, this is like the 2-minute version of the genetics of spinal muscular atrophy. So spinal muscular atrophy is the leading cause of infant mortality in North America. So it's a genetic cause. It's a horrible disease. The infant usually dies within the first year of birth. And it's caused as follows. There's an exon in a particular gene, SMA. That's shown in blue here. Okay. And once in a while, a baby is born with mutations in these 4 locations. It's actually a bit complicated. I won't go into the details. There's 2 versions of the gene, but I'm going to skip that. Essentially, there's mutations in these 4 locations, and that's the reason why the disease develops. Now, if you look at those 4 locations, you might say, well, look at that mutation. It's in an exon. It probably disrupts the protein, and that's what the problem is. Well, that's a synonymous mutation. Okay, so the mutation doesn't disrupt the protein. And what I've overlaid here in orange is the regulatory code that our technique inferred. So the orange blocks are DNA elements that we predict are crucial for regulating the exon, ensuring that the exon is included in the transcript. Okay, so that's what we added, is those orange blocks. And if you compare the mutations with those orange blocks, you see that the mutations do occur in several of these orange blocks. Here's one that's actually within the exon. So the regulatory elements within the exon itself. And it turns out it's the disruption of the regulatory element that causes the disease. So it causes the exon to not be included, which leads to the disease. Okay, so here's a specific example that you can take a snapshot in your mind of, and remember that actually regulatory code, which is what most of us here are interested in, regulatory code is crucial for understanding the genetics of disease. And there's lots and lots of examples like this, and I'm sure many of you know of other examples. So this is well understood, so our technique validates here. We're also looking at the genetics of autism. How many people have looked at the genetics of autism? Raise your hands. Okay, yeah, we've got a few of them, and I talked to Jay about this yesterday or the day before. So if you sequence the genomes of people with autism, that's what I'm showing up here. So each of these is just a toy diagram, obviously. And you compare those genomes to controls, you might hope to see some mutations that differ, okay? And you might think, well, maybe autism has something to do with brain regions, so you might look at genes that have brain annotations, and you might look at other genes, and you might hope to see some patterns, like there's more mutations in the autism cases in brain genes. And if you try to do this, you don't see any patterns. This is sort of a well-known problem. You cannot see patterns in the whole genome sequencing data. So you look at all those mutations, no patterns. And that's, autism is a complex disease. It's not even really a disease, it's a disorder, a spectrum, of course. Who knows what's going on with autism? But here's something interesting. If we take a look at 5 autism genomes, only 5, compare them to controls, and use our technique for scoring these variants according to how much they disrupt regulation, we get a pattern that looks like this. So we actually do see enrichment in autism cases for high scoring variants. All right, so that's what we want to do here as people developing regulatory models. We want to have regulatory models that actually tell us which variants are important, and it's working. Okay, so one slide just to put my work and in context with respect to other approaches. I really don't get genome-wide association studies, I have to admit. It took me a few years to actually really understand what they were doing. And the reason is just because, I've got my perspective, and my perspective is just not matched up with genome-wide association studies. So I think of that as an indirect correlative approach, right? You get a bunch of genomes of people with a disease, people who don't, you look for enrichment. It's like taking a bunch of books that you like and books you don't like, and then looking at a letter and saying, which letter is the right letter here? Okay. So, it's... whereas if you have a regulatory model is saying, this word is important. The word sacrifice means something, right? So the word is important. So mutations in that word are important. So that's what the regulatory model gives us. However, I do, respect other researchers working on genome-wide association studies. and I really do. Okay, so expression and splicing quantitative trait loci. Again, this is a very useful technique for identifying mutations that could be relevant. Like genome-wide association studies, there's the problem of confounding factors like linkage disequilibrium, population stratification, things like that. Okay, so these things can mean that we basically just get an idea of where the mutation is. Maybe it's proximal, and maybe you can use an interesting computational technique to try to find the true mutation. And the talk after mine, Gerald Kwan will be talking about a really interesting method for doing that. I won't spend too much time on this simply because I need to get to my research. So our approach is what I would call a direct causal one. Of course, biochemical investigation is the best thing to do. It's slow and time consuming. And you can identify variants that disrupt protein sequence, so we all know of good tools for doing that. That'll get you sort of 1% of the genome. And what I'm focusing on, and other people here are focusing on, I really enjoyed the talk by Greg yesterday, is identify variants that disrupt regulation. Okay, and we know that regulation comprises a large portion of the genome. We don't know exactly how much, but regulatory elements maybe 5 to 10%, maybe higher estimates depending on how you want to come up with the estimate. So what's our approach? So our approach, and as I said, a couple other people have talked about this kind of approach, so I'm really excited about it, emerging in the community, is basically like this. If we look at a sequence, okay, this is the spinal muscular atrophy case I just showed you. You have a DNA sequence, and I've shown the exon in blue and regulatory elements in orange. You can stick it through what we'll call a regulatory model, which is just a technique that takes in these features extracted from the sequence, and then predicts the expression level. In this case, the splicing level, how frequently the exon is included. So there's two things. There's regulatory code, kind of like software code. The regulatory code resides within the DNA. Now you need an interpreter. The interpreter is a regulatory model that takes that code and makes a prediction for gene expression level, mRNA stability, or splicing, or whatever. Okay. Now, if we look at a mutation, we can just run the mutated sequence through the regulatory model and see what it predicts. If we see a big change, then we think, well, maybe there's a phenotype. Now, maybe the phenotype is benign, or maybe it's a disease. But it gives us a way of ascertaining whether that mutation has an effect. Now, where are these regulatory elements? Of course, there's lots of different places they exist, and people in the room have been looking at regulatory elements in these regions. Today, I'm going to focus on splicing regulations, so intronic elements, splice sites, and exons. So here's our approach, and as Marlis mentioned, it dates back to 2010 when we published a regulatory model of splicing. We take as inputs the reference genome, so no disease here, okay? So this is building the model. Disease is not part of this, no disease. We just use the reference genome, and we use RNA-seq data from healthy human tissues. We extract features from the reference genome, so we look for secondary structures, nucleosome positioning, motifs, all those kinds of things. This is predicted nucleosome positioning, right? Not measured, but computationally predicted. And then we also measure splicing levels. And now we use machine learning. Most recently, we use deep learning techniques to come up with a model that takes DNA sequences input and outputs a prediction for the splicing level. We validate it in different ways using RNA-seq data, RT-PCR, mini-gene reporters. We do knockdown experiments, all sorts of different ways for validation to make sure that the thing works. Okay. So we just use normal, healthy data, if you like, to come up with a regulatory model. But the idea is that what we've learned is basically a proxy for the biochemistry within the cell that is reading the DNA and turning on the transcripts. So now we can apply it to cells that have mutated DNA, cells with variants. So we have different diseases or different technologies that might provide us with variants, but now we extract the variant DNA elements. Same things up here, it's just now we're analyzing the mutated sequence. We apply our regulatory model, just as I showed you on the previous slide. We then score the variant according to how much it disrupts regulation. And now we can do whatever we like with those scored variants. We can combine them with genome-wide association data or other kinds of data. All right, so I'll talk about how we learn the regulatory model. I won't have time to go into many of the details, but I just wanted to give you a sort of a notion for a couple important concepts here. One of them is what I call the principle of invariance. So again, I'm trying to keep things simple. So I'm thinking back to physics. In physics, you say if I throw a ball over here in the air, I'll make the same observation as if I throw it over there. And I'll make the same observation if I do it 10 minutes later. And the principle of invariance allows you to gather information together and build a good model of what's going on, namely that the ball will follow a parabola, no matter where you throw it. It's not always correct. 10 minutes later, maybe the gravitational field has changed. Hopefully not. But it's a good principle, right? It's a strong principle, and you understand what it means. Okay, so that's what we do. So we're going to say that a DNA sequence located somewhere on a chromosome in a human cell, if I were to take that DNA sequence and cut and copy it and put it somewhere else on that chromosome, it would behave the same way. So we'd see the same expression pattern from that sequence across different tissues. Okay, now we know it's wrong because of chromatin interactions, which we've heard about at the meeting, and other kinds of effects. But it's a nice principle, right? Why is it a nice principle? Because it allows us to collect information about these two different sequences and assume that they'll behave the same. So suppose we have two different genes, but they have sequence elements, regulatory features that are nearly identical, then our model would assume that their expression patterns will be similar. Okay, so it allows us to apply statistical techniques. Statistical techniques assume that we have a whole bunch of independent observations of some outcome. And that's what this principle of invariance does for us. So we'll even assume that this is true if we put the gene in a different chromosome, and actually even in a different species. And there's interesting data for TC1 mouse models of cancer where basically you have human genes in the mouse, and you can look at the expression patterns or the splicing patterns. You actually see that they recapitulate human splicing patterns and expression patterns fairly well. All right, so to learn the model, we need to quantify splicing. Most people know about alternative splicing, but I'll just quickly review it here. So here we have our primary transcript. We've got exons, introns. And alternative splicing is a process whereby that exon may be included or may not be included in the transcript. Okay? And what I'm talking about here is based on a population of cells, not a single cell, population of cells. And what we're going to see is some fraction of the transcripts include the exon and some fraction don't. So it could just be due to cell differences between cells, could be due to stochasticity in a single cell, or it could be due to other factors. So we'll measure that fraction using the symbol psi, percent of transcripts with the exon spliced in. Okay, so 0% means we only have this transcript, 100% means we have this one only. And anything in between, of course, tells you the fraction. So what we do for learning is we extract RNA features from a whole bunch of different exons to come up with a data set. So we're going to think of this as IID data, okay, because of our principle of invariance. So for each exon, we extract RNA features. You can just use the raw sequence. We're doing that now. But if you want to be a bit more biological, you can extract things like binding sites of RNA-binding proteins and things like that. Then we have measured data, which again, for each of these exons, we have measured the percent inclusion levels across different tissues. Okay, so that's what this data is. And now we have a machine learning task. Basically, learn to predict these values from these ones. There's different machine learning techniques you can use. We now use deep learning, as I mentioned. But basically, the output is this model that takes as input a DNA sequence, extracts the features, runs it through the model, and makes predictions for the percent inclusion level. All right. There's details, which I won't go into, but you can ask me about afterwards if you'd like. Now, how well does this thing work? So as I said, this is really naive. Amos yesterday mentioned, he took a naive approach, which I think is the right approach. We've got to keep thinking that way. The hope is that we could actually take this model and apply it to new circumstances, genes we've never seen before. Try to use it to actually explain trans effects and things like that. Okay, and that's what we've done, and it seems to work pretty well. So one thing we can do is we can just hold out genes, and we can measure psi, and then predict psi for those held out genes. And this heat map shows you the comparison between our measured and predicted values. So this is what most people in this room have been hoping to do for a long time, right? This is the kind of stuff we like. Our computational model is actually making pretty accurate predictions. The R² is about 0.65 for these cases. Of course, it depends on how you select your exons for testing and all of that kind of thing. We tried to do that honestly. Now, how about a completely independent data set? So this is looking at the RNA-compete data from Hughes and Morris. What I'm showing on the vertical axis here is different RNA-binding proteins. And this has nothing to do with our regulatory model, okay? So the different RNA-binding proteins, and then the affinity of those proteins is computationally predicted for different regions proximal to different exons. Okay, that's what's shown down here. So this is the upstream intron. We just compute the binding affinity of that RNA-binding protein in that region using this in vitro assay. And then what we do is we correlate it with the actual splicing levels of that exon, okay? And we see positive and negative correlations. So these are this just a red block here tells you there's a large correlation between that RNA binding protein and the binding affinity in the upstream intron. And there's different tissues here. There's 16 different tissues. Okay, so some of these proteins are tissue specific, and obviously there's region specificity as well. All right, so that's what you'd expect to see. So that means RNA-compete is working. We're actually figuring out binding affinities of proteins that are predictive of splicing. Now, this is the interesting thing. We can take these splicing levels and subtract off the predictions made by our model. So basically, we're just coming back here and we're taking the measured values, subtracting off the predicted value. Okay, and then we just redo this analysis for that residual data. And this is the result. Okay, so this completely independent assay for ascertaining binding sites of proteins. Most of the information in that assay is already being predicted by our regulatory model. There are some interesting differences, and we're trying to incorporate that to improve our model. We can look at other kinds of measures, like ROC curves and all of that. I won't go into that in detail. Another interesting question to ask is how well do we generalize? We can generalize across different exons. So here's the error rate. So we get an error rate of about 5.5%. That's 1 minus the area under the ROC curve for high versus low, so that kind of thing. We can do things like we train the model using high expression cases where we have a lot of RNA-seq data. We test it using low expression cases. We get still good results, 5.9% error. We can compare across labs. So we can train the data using RNA-seq data from BodyMap, test it using, say, Keisman data, and we get an error rate of 6.1%, still quite good. In fact, interestingly, if we correlate the data from one lab with the data from another lab, we get worse correlation doing that than if we correlate our predictions with data from one of the labs. So our method is actually regularizing the data generated in one lab, producing data that's better. That's why I think that your method might be doing that. Okay, how about different chromosomes? I already told you that works well. Even different species, 9.8% error. So the error is worse. That just means there are different regulatory trans contexts and probably not biochemistry, but different trans contexts in the two species. But the effect of cis elements is quite similar. We've done other things. This was published in 2012 in a project led by Ben Bunko, where we used our splicing code to look at different splicing patterns across 350 million years of evolution. And again, the same sort of thing. It seems to boil down to our regulatory model generalized as well. What's mainly different between the species is the elements in the DNA. Okay, so let's turn to the disease part of my talk. So I've already shown you the picture of how we're going to score variance, right? We're going to run the normal DNA sequence through our model, make a prediction for the splicing level. Then we're going to run the mutated sequence through our model, make a prediction, and see how much it changes. So here's a diagram showing you that, okay? And I just want to define a couple things here. Delta psi is just going to be the average across all tissues of the change in psi induced by that mutation. And there's different ways we can compute delta psi by removing the sign and all this sort of thing. But just think of delta psi as basically the change. It's delta psi, how much the mutation causes the percent inclusion level to change. Then the other thing we can do is come up with what I'll call a regulatory score, which could be like the absolute value of delta psi. But we also use other techniques. Because we use a probabilistic model, we get probability distributions over the levels of psi, and we can use KL divergence and things like that. So there's other kinds of regulatory scores that are just technically better justified. All right, so delta psi and the regulatory score are the two key ways we're going to ascertain the effect of a mutation. So we looked at 500,000 SNVs, including disease ones and common SNPs. And what you're seeing here is the reference psi and the variance psi. And so everything on the diagonal here means that our model predicts that the mutation would not have an effect on the percent inclusion. Everything below means that our model predicts it'll decrease inclusion, and everything above predicts it'll increase inclusion. These ones here, there's a really strong effect over here, where reference psi is close to 100% and the variance psi is very low. A lot of these are due to the introduction of things like stop codons or missense, which could lead to nonsense-mediated mRNA decay, or just missense mutations in general. But you see, what's interesting, you see a lot of points down here, and you see points up here. These are interesting cases. These are cases where the normal sequence from the reference genome is predicted to have low psi, and our model predicts that the mutation causes an increase, okay, a significant increase. So here what I've done is I've organized the data spatially. So if we look at the proximal exon here, and we look at the mutation, so we just map each mutation to its nearest exon, we can see mutations within the exon and outside. So I've sort of mapped all mutations in the genome to this canonical exon here. And you see a few effects. First of all, the blue ones are missense, so you see there's huge effects for the missense mutations, as I mentioned. The green ones are silent, and I really should have plotted the green points on top of the blue. But the green points basically have the same spread as the blue point. So our model predicts that all of these silent mutations have huge regulatory effects. Oh, I should mention these are disease SNVs, okay, not common ones. This does not include common ones. These are disease mutations. What's, of course, on the splice site, so just within the intron, but right next to the splice site, you see large effects. Okay, that's some computational techniques predict those effects. But farther from the splice site, here we're up to 300 nucleotides from the splice site, we're still seeing large regulatory effects predicted by our model. And as I said, these are known disease mutations. Okay. When we compare our technique, I won't have time to talk about this in detail, but when we compare our technique with other methods, there's like a dozen different methods that we've looked at for ascertaining mutations. A lot of the methods look within the exon or near the splice site. But if we look in the intron, we see that our technique, this pie chart here is showing what we've discovered. That's the red versus what other techniques can find. That's the yellow. And then what both techniques find is the orange. Okay, and undetected is shown in the cyan here. So we're finding large numbers of new explanations for these disease variants that other methods can't find. And then similarly within the exon, the splice site is pretty much an easy task. So everyone can do well at the splice site. Now, what about genome-wide association studies? So we were curious what would happen if we took our regulatory model and then applied it to GWAS-implicated SNPs. Okay, so what I'm showing here in black is the distribution of regulatory scores computed using our technique. All right, for non-GWAS implicated common SNPs. That's minor allele frequency greater than 1%. And then in blue is the distribution of regulatory scores computed by our method for the GWAS implicated ones. And then the red are disease annotated ones. These are from HGMD, where there's strong evidence that the variant actually causes or is strongly linked to the disease. And so you see there's a huge shift to the right here in these distributions. So it just means, the take-home picture, one take-home picture from this slide is that our method is certainly doing something quite different from genome-wide association studies. It's adding quite different information. So I think putting them together and combining them is a good thing to do. So what do we learn? What do we learn when we looked at all of these variants? So 650,000 variants, including common and disease ones. We found widespread patterns of mutation-driven aberrant splicing. So it's happening all over the place. We looked at mutations more than 30 nucleotides from any splice site. These are intronic mutations. So they're not directly disturbing the splice site. For those intronic mutations, we found that they alter splicing as predicted by our model 9 times more often than common variants. Okay, so we are explaining disease as a consequence of misregulation due to mutations that are more than 30 nucleotides from the intron, from the splice site. Synonymous exonic mutations are 9 times more likely to alter splicing, so I mentioned this before. And also missense mutations, they score extremely well using our technique, but missense exonic disease mutations that least impact protein function. So when you look at Condell or something like that to try to find mutations that have minimal effect on protein function, we still see enrichment. So it's offering sort of an alternative disease by misregulation hypothesis. So as I mentioned, we're at the point now where we actually think we can have an impact in medicine. So we're really excited about that, and I'm sort of learning about how to collaborate with the medical community, and that's kind of mind-boggling. Like, for example, government regulations. Government regulations, it's very complicated. So in Canada, healthcare is free for everybody. So the problem there is if somebody performs a genetic test and it's really cheap to do the whole exome, they get all those mutations and they discover something else than what they were looking for, they're obligated to follow up on it, right? And so the healthcare costs will balloon, and since healthcare is free for everyone, it's a problem for the government. So currently, it's kind of comical. What people are doing in Canada is they will sequence your exome, or maybe even now there's places that are going to start sequencing the genome. But then what they do is they do what they call an in silico panel, which means they throw all the mutations out except those in the gene they're interested in. That's in silico, yeah. So anyway, they tell me that they're storing those other mutations somewhere, and I'm hoping to get access to those. So we've looked at spinal muscular atrophy, and just to try to get a better understanding of what's going on there. The red mutations are the ones I showed you before that are normal mutations in the disease case. All right. What we did is we looked at the upstream intron of spinal muscular atrophy, and we're wondering, if we could mutate the sequence, could we fix the problem? If we could mutate the sequence, could we get the exon inclusion level to go back up? How would you mutate the sequence? Well, that's another question. There's things like CRISPR, you'd have to have some kind of therapy. There's oligonucleotide therapies that can kind of do this sort of thing. But we just wanted to understand this computationally. So we ran our code across every possible point mutation in this upstream region. All right, so we just computationally predicted what would happen. That's what's shown here. So a mutation here will cause a big decrease in exon inclusion. A mutation here is predicted by our model to cause an increase in exon inclusion. Okay, and similarly over here. We then collaborated with Adrian Craner at Cold Spring Harbor, who has been working on spinal muscular atrophy for quite some time, at least a decade. And he and his students did mini-gene reporter experiments to test the effects of these predictions. And we found that the effects were concordant with our predictions. Modest, not as large as we'd like to have seen, but they matched our predictions. It's hard to tell in the organism what change you need, right? An increase of inclusion of 5% might actually be sufficient. So that requires a lot more further work. We also looked at existing data. People have done a lot of mutagenesis experiments for spinal muscular atrophy in this exon and also in the downstream intron. So we looked at 85 experiments that have been conducted, and on the x-axis here I'm showing the code predicted change due to that mutation. So again, we ran the mutated sequence through the model for a given mutation that we looked up in the literature and predicted the change in percent inclusion. Some of them increase it, some of them decrease it. And then there's the RT-PCR assessed value, which we extracted from the literature. And you see fairly good agreement. The correlation is about 0.74. Okay? So we're able to not just predict whether the mutation is deleterious or damaging, but also predict what direction of effect the mutation will have. Will it increase exon inclusion or decrease exon inclusion? We also looked at different cancers, pancreatic cancer and also nonpolyposis colorectal cancer. What I'm showing you here for nonpolyposis colorectal cancer, 2 genes, MLH1 and MSH2. And in the case of cancer, of course, well, this is a hereditary cancer, I should point out. So in the case of cancer, you know, it doesn't boil down to this nice, simple one mutation explanation, right? So it's a bit more complicated, as you can see from the diagram. So we looked at hundreds of mutations that people have found in patients with the cancer, clinically identified mutations, and that's what each of these stems shows. So each stem is a mutation in a patient. And for each of those mutations, we ran it through the code. There's a whole bunch of exons here in this gene, and what you're seeing for each mutation is the effect that our code predicts it'll have on the proximal exon. So this red stem here means that this patient has a mutation that our code predicts will increase percent inclusion of this exon here by about 25%. All right, now in most cases, it looks like the mutations cause loss of the exon, but there are some cases where our model predicts increased inclusion of that exon. If we compare our data to the RT-PCR data, in this case, it's unlike for spinal muscular atrophy, where a lot of accurate RT-PCR experiments are done for Lynch syndrome, the literature is much more heterogeneous. Some papers just report an effect. They don't really report the size of the effect. And so what we did here is we just binarized the data from the literature and plotted an ROC curve. And we get quite good prediction accuracy for both of these genes. You notice that most of the mutations are close to the splice site, right? It is the case that in these clinically identified mutations, they're close to the splice site. And that's one of the problems we face, and other people have mentioned it in the last couple of days, is ascertainment biases, right? These data sets, whether it's HGMD or whether it's data that has been identified in the clinic, are subject to ascertainment bias. So there's lots of exome sequencing out there, and of course we're not going to find mutations deep within introns using exome sequencing data. We can find some close to the splice site, but not deep. So it remains to be seen what lies further into the introns. And that picture will become more clear as we do whole genome sequencing or mRNA sequencing within these regions. All right, I'm going to finish up by talking about autism, and these are very new results. So this is all in review at the moment. And autism, as I said, is a very complicated thing to analyze. I showed you that toy diagram, right, where when we score the variations, we actually find mutations that are enriched in certain gene categories. So I don't have time to go through all the details of what we did. But we took a look at autism cases. We had 5 genomes, control cases, 12 genomes. We looked at this SNVs, did a whole bunch of quality control steps to extract good ones. We got about 5,000 SNVs from these ones, 10,000 from these ones, computed the regulatory scores using our method. And then what we did is we just identified the genes containing a mutation that our method predicted would have a high regulatory score. So the goal here is to identify genes. And we got 171 genes here, 249 over here. So then this table here basically summarizes what we found. We separated genes into brain genes and other genes. We found 46 brain genes in the autism cases, 125 in the other, among other genes. Controls, it was kind of the other way around, 33 versus 216. And so we did find a sizable increase in the fraction of genes with brain function in the autism cases. So this is new. Most people working on autism wouldn't believe that this is true. So that's interesting. My argument would simply be that we're actually starting to see those buildings, right? We're starting to look at this picture from the right view, and we're starting to see those buildings. So then we decided to not look at brain genes because maybe autism is only partially related to defects in brain genes. We just looked at all functional categories and tried to identify categories that so automatically find which gene categories had enrichment in autism cases. And so that's what this resulted in. So we found a whole bunch of genes with different functional categories. The picture shows the categories that we automatically inferred using our analysis. So we didn't pre-bias this by saying we're going to look at brain genes. We just looked at all Go annotations. But synaptic transition popped up, neuron projection popped up, ones having to do with development and growth popped up. So this makes sense. Out of all the genes we found, a large fraction of them were already known to be associated with autism, but we did find new ones. We found about 20 genes that were not previously linked to autism and that we believe are. So we're scaling this up. We're looking to collaborate with different people who have autism data. We're looking at trio data now, and and gathering data from the internet so we can do a more comprehensive analysis. This is just a small study. All right, so to summarize, I'll just tell you what different people did quickly, or at least just tell you who was involved in the project. So Babak and Leo and Hoy were the main people who did the computational analysis for this work and came up with the models. My experimental collaborators are Ben Blenko on splicing assays and Steve Scher on autism, Adrian Craner down here on the spinal muscular atrophy work. Josef Barash was involved in the original derivation of the code, and he's been participating to some degree in our disease analysis work. Quaid Morris and Tim Hughes and people in their lab for their help with the RNA-compete analysis. And that's it. Thank you.
Brendan, thank you very much for a beautiful, beautiful talk. My question is on the very, very last part on the autism spectrum disorder. So you would expect that some of these mutations, when you put them in the context of the cell, are sort of pushing the brain in one direction, and other mutations are actually pushing the brain in another direction. So my question is, currently your score is simply just counting up overall dysregulation, if you wish, of every gene, assuming that there's some sort of perfect level from which we're always going away.
Sorry, what was the last sentence?
Assuming that there's some kind of perfect, level for every gene from which we're going away. But instead, if you start thinking about directionality of effect, and you start sort of putting these genes in the context of a regulatory network, you will then find mutations that are pushing you away and others that are actually bringing you back. Are you, am I sort of...
No, that's right. That's okay. a very good comment. Yeah, it comes back to keep it simple. I'm trying to just do the best job I can of a simple cis element view of what's going on. But you're totally right. It would be great to combine this with models that are taking into account more what's going on in sort of in the cell state. I think of that as like the cell state.
Yeah.
As you said, mutations that push you into some other cell state, but other mutations that bring you back.
Exactly.
Yeah.
Basically, if there's a patient who comes in, you want to look at their whole repertoire and say,
right. Yeah.
Here's what I'm predicting.
Yeah.
The related question is, if you have gene expression profiles for that individual, you may not have them in brain, but you may have them in blood, for example, where, the cis code is the same regardless of the tissue. You could basically start asking about dysregulation of splicing, which is actually instantiated in addition to predicted by your model, and then look whether your predictions are in fact reflected in mis-splicing events. Would that sort of fit within your same model, or?
Yeah, we can. So you're just talking about sort of specific effects in different cases that are caused by changes in other aspects of what's going on, like changes in trans factors or something, regulatory protein, changes in regulatory protein abundances.
Of course that's important, because then you're sort of looking at the global effect of each of those regulators.
Yeah.
But what I was thinking is that every time we see a mutation in a gene or in a splice site, we're sort of predicting that that mutation is going to have an effect, for example, in splicing. You can then go and look at RNA-seq and say,
oh, I see. Yeah.
If I now sort of filtered my predictions through what's actually happening in a surrogate tissue, could I do better in the prediction?
Yeah. Well, that's a good idea. And we do that, actually. Okay. And it does work, yeah.
Great.
Thank you, Brendan. It was very enjoyable, and the talk came in a very timely manner for me, because last night after Ellen Rothenberg's talk, I went to my room thinking, with my machine learning that I'm doing, I'm never going to be able to learn any biology. And you bring us back to show, no, we can learn some biology doing our data analysis. That's great. You brought back the hope.
Good, I'm glad.
So, very simple technical question. The sequence information that you're learning is with kernels? I'm sorry, with k-mers?
That's part of it. That's the large fraction of the features, but we also compute things like secondary structure.
Okay.
We compute nucleosome positioning.
Okay.
Lots and lots of features.
Okay.
Yeah.
So my question is, at what point do you decide that you have to go deep in your learning? How did that come to happen?
Oh, sorry, what was that last question?
Why did you have to go into a deep network?
Oh, I see, right.
At what point do you make that decision?
Yeah. So actually what we're trying to move towards is more of a system where we can look at the raw DNA sequence. looking at these features is important because it allows us to trace back to the biology, but there are unknown regulators, and so we're more looking at the raw sequence data now. And when you look at the raw sequence data, you need to really worry about overfitting because there's just so many nucleotides, of course. Yeah, so that's why we got into deep learning. Okay. Yeah.
Hi. Thanks. That was a wonderful talk. I was wondering if you've looked at any of the features. So you have your set of these SNPs. I was wondering if you've looked at the features, like the allele frequency spectrum, and specifically whether they tend to segregate differently across populations.
So we do look at the features. One reason we've looked at the features is to try to identify, of course, which, say, protein regulators are responsible. Another one is to try to understand whether there's combinatoric interactions between them, and we do see that. Another one is for validation. So one thing we've done is we've looked at features that are likely the binding sites of particular regulators like MBNL, and then we can look at knockdown data. So we look at data where we've knocked down MBNL and see if our model makes the correct prediction. And we find in that sort of case we get quite a strong signal, so we're able to predict the effect of it. In terms of stratification, no, we haven't looked at that, but it's a good idea. Yeah, that'd be good.
Thanks for a great talk. This is another question about looking at features. In your deep network, have you looked at what the hidden nodes are picking up? And are they picking up things along the lines of what your previous methods were finding, or are the hidden nodes finding something you hadn't thought to look at before?
Yeah, that's a good question. It's hard to interpret what's going on in the neural net. What you can do, and I think this is very cool because it allows you to use the neural net as a sort of a surrogate for the system, is you can ask questions like, in a particular exon, there's a binding site for one protein and a binding site for another protein. If I turn both of them off, what happens? If I turn one of them on and one of them off, what happens? Or the other way around. And so you can look at the combinatorics of interaction just by simulating what's going to happen by putting it through the neural net. And we've done that, and we find really interesting sort of and type and or type interactions between features. So I kind of think of it that way. I kind of think of it now we have a system where you can ask it anything you'd like, and we sort of have the computational simulator of what's going on, and it'll tell you what the interaction looks like. And then you could go and test it in the lab using a more directed experiment.
Thank you.
Yep.
Brandon, thanks for the great talk. I think you partially answered my question, but I was wondering about the complexity of your predictive model. How can you consider the combinatorial effects of different mutations or some nonlinear effects of these variables in your model?
Sorry, I understand what you're getting at, the combinatorial nonlinear effects,
Yeah.
but what's the actual question? What are we?
Do you consider them in your model, or?
Oh, yes. So the neural network can learn about combinatorial interactions basically by figuring out, like, there's hidden nodes, and the hidden nodes have certain weights connected to the feature detectors, and then the unit can sort of fire if there's an and type combination of the two features, or not fire if there's just one or the other present or neither one is present. So yeah, we do learn about the combinatorics. Does that answer your question?
Yeah.
Okay, great.
Thanks.
Our next speaker will be Gerald Kwan, who